Hey folks, I'm here to talk about carbohydrates now. Okay, this is our second category of biochemicals. We talked about the hydrocarbons first, which aren't so important to our biochem talk, but carbohydrates, personally, they're my favorites. I'm sure there are a lot of your favorites too. So let's get started, all right? Carbohydrates. They obviously include the sugars and all the polymers of sugars. What does that mean? When you think of carbs, you probably think of like starchy foods like breads and pastas. Yes, that's more the complex ones. Sometimes you don't think of sugars as that. You think of them as another category, but no, they're all in the same category. Carbohydrates, what are they? Well, they have this molecular formula of like CX H2OX. In other words, however many carbons there are water molecules. That's why they call it a carbohydrate. Carbon hydrate of carbon. And carbohydrates are our immediate fuel. They're the ones that our bodies are going to turn to first for energy. They're not going to turn to fat first. They're going to turn to the carbs. Carbs, I like to consider them like the sticks and twigs and paper and even the dry logs you would put on a fire. Okay, and they're built up in the chloroplasts, but yet they're broken down in the mitochondria. So when it comes to cell parts with carbohydrates, chloroplasts are going to be making them, mitochondria are going to be breaking them. Okay, let's move on to our next slide. Our next slide is the first group of carbohydrates called the monosaccharides. You usually find them ending in ose, glucose, galactose, fructose. A lot of them end in ose. Ose usually means sugar. Simplest carbohydrates are the monosaccharides. They're the single sugars. I like to call them the one-ring sugars because often we see them in the ring form, which is here. But they can be in a linear form as well, okay? And the most common of the monosaccharides are the ones that are the C6H12O6, which happens to be glucose. But as we'll see later, monosaccharides may have different monosaccharides, but they all may have the same molecular formula. You might be thinking, oh, sounds like an isomer. Okay, just talk a little bit about glucose here. And these are three different forms of glucose. We've got the alpha form, the linear form, and the beta form. What's different here? Obviously, the linear form is obviously the most different because it's not in a ring form. But what's different about this one and that one? Okay? If we look at these OHs, these little hydroxyl groups, and I'm going to be mentioning functional groups a lot now, like a double bonded oxygen to a carbon means a carbonyl. All sugars have a carbonyl group if you look at the linear form. That carbonyl group is bonded up in the ring form. But these little OHs, those hydroxyl groups, when it goes and we read right to left when we look at this, down, down, up, down. Okay, that is the classic glucose molecule, down, down, up, down. But over here we see it goes up, down, up, down. That's also glucose. And you might be thinking, but that's like a different covalent bond. That's a different type of structural isomer here. It actually tends to be the same thing. Yes, it's alpha. Yes, it's beta. Alpha down. Beta is up here. But it does still perform the same functions in either form. Okay, but we're going to see where that comes into later when we get into like the polysaccharides. Okay, remember mono means one. Okay, monosaccharides. They're classified by not only the location of their carbonyl group as an aldose or a ketose. Remember your aldehydes and ketones. Okay, well aldoses are those sugars, and ketoses are sugars, but where is the carbonyl group? So what is this one here? But it's also classified by the number of carbons. So let's skip to this next slide. This is really going to help us out a lot when we talk about this. Okay, so look at aldoses. What do you notice about the carbonyl group in all these linear forms? Yeah, they're at the end of the chain, okay? Just like aldehydes should be at the end of a chain, while aldoses, the carbonyl group should be at the end. So the ald comes in with aldehyde, but we put ose because we're talking about sugars, okay? Then ketoses, notice the carbonyl group is in the middle of the chain, just like ketones should be. So that's the first classification. Ketones, it was in the middle, the carbonyl group, and if you're forgetting a carbonyl group, it's a carbon with a double bond O. Look at your chart of functional groups, and you'll remember that. And aldose is when we have a carbonyl group at the end. But to further classify them, see trioses, pentoses, hexoses. Trioses have 3 carbons to them. Pentoses have 5 carbons. This is your ribose and your deoxyribose, which are the sugars that are in DNA and RNA. And then the most commons are the hexoses. Those are the ones with 6 carbons. Your glucose, galactose, and your fructose. Okay, notice glucose and galactose are types of aldehydes or aldoses. Okay, because the carbonyl group is at the end. But fructose, it's in the middle. Remember, this is all in the linear form. And sugars can take on the linear form or the ring form. All of these can in different kind of situations. A lot of times heat. So as soon as like a sugar gets into your body, it changes from a ring form to like a linear form. So heat is one that gets it out and into this linear form. Okay? So hopefully we're not having any questions, but one more time, ketoses are like ketones because this carbonyl group is in the middle. Okay, so we'll put this at the side. Carbonyl group, carbonyl in middle. Okay, and aldoses, the carbonyls at the end. Carbonyl, sorry, my hand gets in the way at first, at end. Okay, and we both say oses for these because they're sugars. Okay, triose 3, pentose 5, hexose 6. Okay, that is an important slide there, so make sure you pay attention to that. Okay, next slide. So now we're going to move on to the disaccharides. Mono was 1, di means 2. Okay, so we're going to go mono e mono, make di. Okay, 1 plus 1 equals 2. Now you would think if you took like 2 hexoses, right, C6H12O6 plus C6H12O6, a disaccharide should have the formula C12H24O12 because you just double them. But why do I subtract water here? And that goes back to that whole dehydration synthesis, okay? When you got to put 2 monomers together, and you know what I'm going to get here, okay? I'm going to get my beads out, okay? So if we take, say, this is one monosaccharide and this is another monosaccharide, and we want to link them together just like in the previous forms. When we link them together, we have to remove a water molecule. Okay, so that's why we removed 1. And there will be one molecule, one water molecule removed each time. So here's a little riddle for you. Okay, if I have this long polymer, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 monomers here. How many water molecules were removed to link them? So you should have answered 11. Okay, so 11 water molecules had to be removed each time these link up. Okay, so getting back to our disaccharides, so if we add 2 monosaccharides together, we need to subtract 1. Add 3 together, we would need to subtract 2. So the formulas for all disaccharides are C12H22O11. Those we took 2 hydrogens and an oxygen away to get that. Okay, now disaccharides are a little more common. Hopefully not this first one though. Maltose is the kind of sugar you find in a lot of liquors, okay, especially your hard liquors and stuff, you know, like your gins and your whiskeys and stuff. But maltose, malted sugar, you may have heard that in like Ovaltine, I think, has malted sugar in it. Malt sugar isn't what has the liquor in it, but it's often associated with liquor things. So we sometimes call maltose that liquor sugar. Okay, lactose, I'm sure some of you are common with it. I'm sure some of you might not even be able to digest it very well. Okay, and later on in the year we'll get into like why we can't digest and why actually it was an evolutionary advantage to be able to digest lactose. We shouldn't be able to really digest it. And you can see maltose was a glucose plus a glucose, of course minus that water. A glucose plus a galactose would make lactose. That's why they get the name lact from the galactose in it. That's your milk sugar. And a glucose plus a fructose is sucrose. That's good old table sugar. You know, maybe you sprinkle cinnamon sugar and you mix cinnamon with sugar and you sprinkle it on whatever you would sprinkle it on. Okay, now whenever we bond these together, we know it's dehydration synthesis, but in first year bio, you never gave the bond a name. Like when we bonded amino acids together, we talked about the peptide bond, but we never gave the bond a name in first year bio. So when we link a monosaccharide to a monosaccharide, in other words, when I have my disaccharide here, what is the name of this bond? Well, it's called a glycosidic linkage. Yeah, it's a complex word, but it's just another word that you got to understand. It's the bond between monosaccharides and a polysaccharide. So speaking of polysaccharides, that's our next category. We had mono was 1, disaccharides are 2, and now, wanted to show you another dehydration synthesis here. Here's the formation of maltose, you can see. Here's a glucose, and you can see the H from one of the hydroxyls and the OH, the whole hydroxyl group, will combine, okay, to form the water that's removed, and then here is your glycosidic linkage, okay? Here is your glycosidic linkage from here to here, see? That we call a 1,4 glycosidic linkage because it's number 1. Remember, we go left to right. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4. So it's a 1,4, the 1 to the 4 glycosidic linkage. Okay, that happened, and it happened via dehydration synthesis. Here is glucose and a fructose, and when we look at this, and if we count the carbons, 1, 2, 3, okay, 4, oh, excuse me, I'm counting that wrong there. Anyway, let's go move on here. H, OH, take out the water, there's your glycosidic linkage. Why is it called 1, 2? Because fructose is different with how they number it, and that's why I wanted to get to this picture. Even though this is number 1, 2, 3, 4, this is our carbon number 1, this is our carbon number 2, because fructose is, remember, it was in that category of ketoses, so it's reverse order for ketoses. Not too particular about this 1, 2, and 1, 4, so don't let that stress you out, the 1, 2, and 1, 4. All I really want you to know is that from dehydration synthesis, we form this glycosidic linkage, and that's what it is. And look at what I wrote down here. Shape matters, okay? Because the shape of these molecules are going to matter as to whether, like, how sweet this one is. Sucrose is much sweeter than maltose. This is a dry kind of sweetness. This is like a very sweet sweetness. Okay? All right, now we get to move on to our polysaccharides. So polysaccharides, poly means many monosaccharides all linked together by those glycosidic linkages. And there's 4 polysaccharides in particular that we're going to talk about. Starch, glycogen, chitin, and cellulose, okay? And the difference with these we're going to see in the picture, okay? But first, just a little bit. Starch, a storage polysaccharides in plants. Plants will store extra starch as granules in their chloroplasts and other plastids called like leucoplasts. Glycogen is a storage polysaccharides in animals, so storage in plants, storage in animals. We find a lot of glycogen in our liver cells, okay, and some in our muscle cells, but a big supply in our liver cells, and that's where we really go for it when we have our fight or flight. When we get startled, we really kind of bust out that glycogen in our livers. Chitin is a structural polysaccharide. This is found in the exoskeleton of arthropods, in other words, bugs, but also it's in the cell walls of many fungi. Okay, in the fungi, it's more like a leathery texture, but in arthropods, the exoskeleton is very like firm texture, hard texture. Okay, that's why a bug goes crunch when you step on it, but yet a mushroom goes like when you step on it. Okay, crunch for the chitin in an arthropod shell, but leathery kind of feeling, and that's good because I wouldn't want to crunch on mushrooms, like, you know, if I was eating mushrooms like on a steak or something. Okay, and finally, the last polysaccharide, cellulose, better known as fiber. It's the component. It's a major component in the cell walls of plant cells. Okay, and we can't digest those cell walls. Okay, we cannot digest cellulose because of its toughness, its rigidness, and we're going to see the difference in the two types of forms of these four polysaccharides. Okay, so we're getting down to the nitty-gritty here. And starch and cellulose, okay? Here's what I always look at. Starch has what we call a 1,4, okay, a 1,4, like linkage of alpha glucose molecules. Remember those glucose molecules? I can flip back to that page way back in the beginning. We had the 3 types of glucose molecules. I'm looking for it. I found it. Okay, this was the alpha glucose, this was the beta glucose. Okay, so going back to that, when we make starch, it's a number of alpha glucose molecules. These alpha glucose molecules, you'll notice have all their hydroxyl groups going down, 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 down. Okay, that's starch. What is that going to do for starch? We'll see in the next thing. But when it's beta glucose molecules, you're going to see the up, down, up, down, up, down. What does that cause? Okay, it caused starch to be able to be digested, but cellulose is unable to be digested. Okay, except by like certain things where, like termites, they have these symbiotic microbes, these little organisms that live within their stomachs or their guts that are able to digest that cellulose. Okay, it's not really the cow that can do it. It's not really termites that can do it. It's actually the little organisms that live inside them that can do it. We always say cows can digest cellulose. It's not. They have these things living in their digestive tract. If we had them in there, I guess we would be able to digest cellulose. But cellulose is really not digestible in our tracts, and so it works like a steam shovel, just pushing the poop out. Okay, all right. And what do these alpha linkages and beta linkages, what does it mean? What does it happen? Well, starch will tend to have this helical structure because of that. Because they're all like that, I think it goes, and it starts to coil, and it's much easier to digest. That's the helical structure of starches, okay? But with cellulose, it doesn't coil. Instead, and this was the best picture I could find, because it has that up, down, it forms these pleated sheets. And those pleated sheets can really pack tightly and form a very tough structure that we can't break down. We can't digest these bonds between them. So that's enough about our carbohydrates for now. Chitin and, what was the other one? Chitin and, I'm forgetting. The other one that I wanted to talk about, chitin and glycogen. We're not going to get into the structure. I really want you to know the difference between cellulose and starch, but chitin and glycogen, you just need to know their purposes. And that's it for carbs today. Okay?